Good evening or good afternoon if you're in the UK. Welcome to our members and our guests to this evening's talk, Neutralising the Threat of COVID-19. We are delighted to welcome Professor William James from the University of Oxford to talk about his research in virology, in particular his work on the therapeutic treatment of COVID-19. Even with the vaccines, we know that people will still get sick and sadly some seriously. And therapeutics remain a key part of the multi-pronged strategy to tackle this virus. Before I introduce our guest and our moderator, I'd just like to say thank you to Dr. Elizabeth Lee, the CEO of Sunway Education, for connecting everybody together for this event. Please post your questions in the YouTube chat, and Melissa and I will take them forward into the studio for Professor Abhi and Professor William. So please let me introduce our speaker for this evening. Professor William James, Professor of Virology, has run a research lab at the Dunn School in Oxford since 1984, investigating the biology of viruses, including dengue, Zika, Bonamwera, HIV, and recently the SARS-CoV-2. In parallel, he's tutored medical and biochemistry students at Teddy Hall, Christchurch, Exeter, Magdalen, and since 2001, Brasenose College, where he is the Geoffrey Cheah Professorial Fellow and Tutor. Between 2011-2017 inclusive, he was seconded to be the Senior Pro-Vice Chancellor of Oxford, responsible for institutional strategic planning and budgeting. He's the director of Oxford's James and Lillian Martin Centre for Stem Cell Research and head of the core virus containment facility for Oxford's COVID-19 research efforts. His particular research focus during 2020 has been devising and evaluating agents that neutralize the infectivity of the SARS-CoV-2, resulting in high-impact recent publications. Before I pass over to our moderator, I'd just like to say a few words about him. Many of us already know him. Professor Abhi Veerakumarasivam is currently the Dean of School of Medical and Life Sciences at Sunway University, having started his academic career at University Putra, Malaysia. His research focus is in cancer research and in recent years has spent significant time promoting science leadership and communication in Malaysia and in the region. He's the current chair of the International Network for Government Science Advice Asia and co-chair of the ASEAN Young Scientists Network. So welcome to Professor Abhi, and especially welcome to Professor William. Really looking forward to this talk. Please do post all your questions in the chat. I'm sure we're going to have a lot of them. So over to you, Professor Abhi.
Thank you, Emma. Emma is the president of the Oxbridge Society, and I thank the Society for getting this, various different forums out there to not inform inform, but hopefully entertain. And today, of course, great to again have Professor William James. Although we would love to host you in Sunway, just like what we did in 2016. That's the first time I met you when you did this amazing lecture and demonstration on macrophages and infection. So the expectations, I'm sure, are really high, and everyone's on here not to listen to me. So without further ado, let me present Professor William James and his lecture for today.
Thank you very much, Abhi. It's been a great pleasure to be invited, and it's been so nice in the past to visit Malaysia and Sunway and my friends in the other universities in Malaysia. And it's a great sadness that I haven't been able to come over the last couple of years. So let's hope that all of this current panic will pass over the next 6 months or so, and we'll be able to put that right.
That's right.
So I'll start my talk now. So I've called my talk Neutralising the Threat of COVID-19. And this is a very recent picture of the virus particle as revealed by something called cryo-electron microscopy, just to give you an idea of what it looks like. So to neutralise the threat of the virus, we have to be able to intercept its replication cycle. So it's important to understand a little bit about the virus itself and how it spreads from person to person. And here, that's that picture of the virus that I've shown you, the real picture of the virus. And this is a cartoon we often show of the same components. You can see they are quite quite similar. This is what we understand to be the virus. It has an envelope with some bad news in it. That's RNA, the message that will make virus proteins. And the envelope is studded with a variety of proteins, particularly this spike protein here, which is responsible for the virus getting into cells. Now, it's a very small thing, 0.14 of a micrometer, extremely tiny. And it's transmitted by a variety of routes, but the most important route appears to be small droplets, which we call aerosols, less than 5 microns, typically about 1 micron size. And it's often a good way of thinking about how these droplets behave is to think about the way that smoke behaves. If you have friends or acquaintances that smoke cigarettes, you know how smoke drifts around and gradually floats further and further afield and doesn't just drop to the ground within 2 meters necessarily. But in a well-ventilated place and plenty of space, then you're exposed to less of it. And if you wear masks, less still. So I've reduced the scale here so you can see the little virus particles in their tiny little droplet. And if we reduce that scale still further, you probably can't quite see these tiny little dots, blue dots here, which are the aerosol droplets that have got into the airway epithelial cells. And this is a little cartoon of airway epithelial cells. And now we're down at the 10 micron stage, not at the 0.14 micron stage or the 1 micron, but down at 10 microns. So we're really, really tiny now. And these are the interactions between the viruses in these aerosol droplets and the epithelial cells that give rise to infection. Now, the virus itself spreads from person to person and replicates mostly in the upper respiratory epithelia, the nasopharyngeal tract, and often doesn't generate symptoms, or more frequently a generalized febrile condition with sore throats and so on. And as I've said, it's spread by aerosol, where inhaling such tiny aerosols results in infection of those epithelial cells and their replication and so on and so forth. The real trouble occurs when the virus then descends into the lower airways and infects the cells of the air sacs or alveoli, which are the vast majority of the tissue of the lungs down here. And once the virus gets down into this zone, then we can have very severe symptoms, including hypoxia and pneumonia, pneumonitis, intravascular coagulation, and many other symptoms which are familiar to people, which can result in death if not treated well, or even sometimes if treated well. So what we want to be able to do is to prevent one of two sorts of steps. Either prevent the transmission of the virus from person to person through those aerosol droplets, the very early stage of virus getting into the upper airways, or prevent the descent of the virus into the lower airways where the really severe symptoms take place, or preferably both of those things. How can we go about doing that? Now I'm zooming in a little bit here, and I'm going to show you just a little bit of data from a paper that we recently published or submitted on the right-hand side here with Pei Zhang in Oxford, of what's going on deep down inside the cell. This is a real snapshot of a virus particle being produced inside a cell, and it's represented on this side of the screen, if you can see it, by these structures here. But at this stage, it's really hard for most agents of the sort we're going to talk about in a moment to get at the virus. Drugs can get at the virus, but antibodies and similar agents cannot. They have to intervene when the virus particles are coming from the aerosol droplets and or from other epithelial cells in the same host and binding to the receptors on the surface of these epithelial cells. So this is the surface of the epithelial cell right here. This is the inside, and that's the outside. And this is a virus particle bound to the receptors on the cell surface before it enters the cell. Once it's entered the cell, it's no longer available for antibodies to interfere with the process. So what we're looking for are antibodies and similar molecules, and these Y-shaped molecules here, to prevent this process. Well, are we able to do that? There are two major ways we can do that. Oh, so before I do that, there's a very cute movie I'm going to show you from my colleague down at the Crick in London, Donald Brenton, who has produced this movie of the spike protein doing its business. What's going on here is the spike is lifting up its little heads and binding to the receptor on the surface of the cell. We're looking at the surface of just one spike protein bound to here 3 receptor molecules. And it's that interaction and those changes, the movement up of those domains and the binding to the receptor, that we need to be able to prevent. So we can do this in 2 main ways. I'm going to give you a little digression because I know what is on the surface of most people's minds at the moment is how are the various vaccines that are now being rolled out going to be useful. And so I will just give a little digression on those to put things in context. So all vaccines are intended to induce an active state of immunity in a person so that they can't be infected or get serious disease from the virus. So very briefly, these are some slides very kindly provided to me by Sarah Gilbert and Per Alfredson from AstraZeneca a couple of weeks ago on the then state of their vaccine rollout. This slide is our own data from my lab on which was published in The Lancet back in July, work we did on the phase 2 trial of the safety and efficacy of the Oxford COVID vaccine at that stage, which essentially enabled them to go to the phase 3 level, which showed whether you get protection from disease or not. And it's these numbers that matter. Now, I'll try and explain them. These are the sorts of data we generate all the time in my laboratory. They show if you dilute the antibody from somebody who's received immunization by 10-fold here, 100-fold here, or 1,000-fold here, you get lesser and lesser degrees of inhibition of virus infectivity. This is when they're enrolled. This is 28 days after they've been immunized for the first time, and this is a couple of weeks after they've been boosted with a second dose. And you can see the shapes of the curves change, I hope. You can see some really sharp curves here post-boost. And we then try and measure, we fit these curves to a particular equation that measures the concentration of serum, which gives us this 50% point here. We plot that, all of these data on a graph like this. And what we can see is that the strength of the antibody responses in vaccine recipients rises with time from when they're enrolled to when they've had their second dose of vaccine. But there are a couple of things to say about this, which gives some insight, I think, into the results of the phase 3 trial. And that is not everybody responds in quite the same way. So of the only 6 people we're seeing here that had the boost, 4 of them produced good, strong, neutralizing antibody responses that went up very strongly after the boost, up to levels that we would expect to protect them from infection. But 2 didn't rise significantly from the priming stage. They remained at the level of immunity that they gained from the priming first dose of the vaccine. And we don't know whether these 2 individuals would be protected against the infection. They might or might not. But if not, it might explain why only something like 2/3 of the people that received the boost were indeed protected. It's a very small sample here, of course, but knowing that at least the majority of people with a boost got a very strong antibody response that neutralized virus infectivity gave them the confidence to go ahead with a much larger scale trial of tens of thousands of recipients in the phase 3. So what AstraZeneca have been doing is a 4-fold approach, working with governments to get funding to expand access to that vaccine, working with development agencies to expand clinical trials and accelerate approvals, working with multilateral organizations to ensure fair allocation and distribution of those vaccines, and supply partners who can make vaccine to increase production and distribution. And really this latter thing, I think, has been rather strikingly successful, with production being of this adenoviral vaccine being racked up in multiple countries around the world. And the most impressive for me is the Serum Institute of India, which is about to supply just over 1 billion doses of this vaccine, principally for low and middle income countries. So because it's a relatively straightforward vaccine to make and quite cheap and easy to transport and store, it's much easier to generate the sort of 3 billion doses that they're planning here than for some of the alternative vaccines. And you will know that the phase 3 trial has been reported, showing above threshold levels of protection, but there are still questions about what dosage is the optimal dosage. So these phase 3 trials are going on in multiple countries around the world to get more information on that very question. So I'm back to the sort of work that we've been doing since that little study we helped the vaccine crowd with back in June. We've been looking at an alternative approach to generating immunity in people, a passive system where we could use immunity as a therapy, as a treatment, rather than as a preventative. Can we harness the power of antibodies and similar molecules to treat infection if we catch it early enough? So I'm not going to go through all the data we've generated on this. Is a single data slide summarizing one little snapshot of some of the agents we've looked at. Now, the scales look similar, but actually it's slightly different from the ones I showed you before. Here we're expressing these agents in concentration terms, in nanomolar terms. So they're very, very low concentrations of some of these agents that give you very strong efficacy at neutralizing virus. And these are the concentrations that we're finding that give you typically neutralization of virus for 3 different classes of agent. Antibodies are things that nature makes in your body. And the very best antibodies we've looked at have, which are dimers by evolution, have potencies at the 0.25 nanomolar range. But when we break them into their respective monomeric halves, they're about 1,000-fold less potent than that. Nanobodies are a type of antibody or derivative of a type of antibody made by camelids, that's llamas and alpacas and things like that, which are able to be engineered to be very much smaller and cheaper to make and much more stable than antibodies. And we found nanobodies that are able to neutralize at about 10-fold better potency than the antibodies. And when we cut these into their 2 separate parts, we get again about 1,000-fold worsening of their effectiveness. So it looks like something of a rule in neutralization that being a dimer, as opposed to a monomer, gives you about 1,000-fold improvement. A final class of agent that we're very excited about called aptamers. These are completely synthetic. You make they're like antibodies, but made of nucleic acid, and you don't use cells, let alone animals to make them. They're entirely chemically synthesized and selected. The best monomeric aptamer we found so far has an effectiveness at 1 nanomolar, which is much, much more potent than any other monomer that we've seen so far. So the question will be whether we can move its potency up by 1,000-fold when we make it a dimer. So we're very excited about the possibility of using that. So finally, how would you use any of these agents in practice? Well, you already know, I think, that a certain former US president has had a very large dose of antibody, the Regeneron cocktail of antibodies, administered at a very early stage of his infection, and he recovered surprisingly quickly. We don't know whether we can ascribe his recovery to these antibodies, but it is at least possible. Now, those are large molecules. This is the size. I've tried to draw these molecules to scale over here. That's the size of an antibody molecule, IgG. And they're too large to use as ordinary drugs. So you have to introduce them into the blood via a drip. And you have to give very, very large amounts of them. He received 8 grams of such antibodies, and we think that that would have cost about $150,000 for his dose, which is not really the sort of scale of cost that we could roll out around the world to everybody that has the virus. Costs may come down quite a bit, but not by too much. The question really is, could we use other modalities to introduce agents that were much cheaper and much smaller and more robust, such as the nanobodies I was talking about before, which you can see are much smaller than the antibodies, or these aptamers that are smaller still and cheaper still. And we're hoping to get to the stage where they can be introduced by very cheap over-the-counter inhalers of the sort that are already used widely for people with asthma and so on. And in the laboratory, what we're trying to understand is exactly what the mechanism is by which these dimer agents are so much more potent than the monomers in this inhibition of virus binding to cell receptors and infecting cells. So that's the sort of science interest, and this is the applied interest that we have in our laboratory at the moment. So with that, I'll just very briefly acknowledge the very large spectrum of people that we've been working with since the spring. Becky Moore is my right-hand woman in the containment laboratory. She runs the laboratory for me. We were a team of just 2 for about a month, running all of the projects in the containment level laboratory, but thankfully we've managed to train up a large team of people since then. Adam runs the virus work in my lab and is assisted by Javier and Michael. And we have lots of Oxford collaborators and collaborators from other universities around the world and indeed companies, without whom none of this work would be possible. So at that point, I'll finish my presentation and take questions. Thank you very much.
Wow, William, thank you so much for an amazing run through from what viruses are, what do they do, sharing a little bit of the insights in terms of how the Oxford AstraZeneca vaccine was developed, and finally, your really exciting work in neutralizing antibodies. Everyone listening here, all you need to do is put William James, Oxford, and there's an amazing body of work, including some amazing videos through the Oxford series that you can also check on it. But as you said, I think today, many are on here today to also find out what does the eminent virologist think about the current landscape with vaccines, and also in terms of the different waves that we are experiencing, and how might we actually find a route to this idealized post-normal world that we're looking forward to. So please feel free to type in your questions. We have Emma and Melissa who will be very kindly giving me these questions, and I will be very happy to read them out. So do not hesitate. So before I even start my question, there's a question from Professor Maha Abdullah from University Putra, Malaysia, whose question is, was there a positive association of in terms of its neutralizing efficacy with gender? Was there any gender association?
On the samples we looked at, they weren't big enough to see that, but I haven't seen that broken down for the phase 3 trials yet. I'm sorry, I can't answer that. There could easily be differences by gender and by many other categories as well, and that will all come out with the analysis that, as you know, hasn't yet been fully published.
Sure, great. So while we wait for more questions, my question is, I think, based on the WHO's latest draft landscape of COVID-19 candidate vaccines, there are currently about 51 candidate vaccines in clinical evaluation, and at least 13 have registered phase 3 clinical trials.
Yep.
A few days ago, the UK granted an emergency authorization for the COVID-19 vaccine made by Pfizer and BioNTech, making it the first Western nation to approve a COVID-19 vaccine. China and Russia, of course, had previously greenlit other candidates, but perhaps without this large-scale testing. So, William, could you perhaps share with the audience the distinct and subtle variations that exist between the major candidate vaccines, and why you think some will be more successful targets than others, appreciating your biases for the Oxford vaccine?
I should have said this. We should do a conflict of interest statement at the beginning. I have no conflict of interest in any of these things, unlike some of my colleagues. So it doesn't matter financially to me one way or the other which vaccine works. So yes, it's a very good question, Abby. I think if we take it from the top, the most old-fashioned and simplest form of vaccine for viruses is to take the actual virus itself and inactivate it with formalin or some other agent like that. And that's how the original Salk vaccine for polio was made, and that's how most of the influenza virus vaccines are still made. Now, in the world of SARS-CoV-2, there are two such inactivated vaccines I know that are now at very advanced levels, in fact, being rolled out, and they're both from China. So Sinopharm's product is of that type. We know this sort of approach works pretty much predictably. Obviously, they've done their trials according to their own regulatory authorities, and they've passed all the efficacy and safety barriers. So I'm not at all surprised that the inactivated virus vaccine approach that Sinopharm are using works to provide protective immunity. The downsides are, and they're rolling it out very wide. I think there have been 1 million doses already issued, according to the last reports I saw, and 10,000 I think are being supplied to Malaysia, which is very nice of them. I think it's for frontline workers or something like that. The downsides of that sort of vaccine are really the first one is cost. It's actually hard work to generate purified virus particles in sufficient numbers to inactivate them and still have them immunogenic. But they've clearly got that cracked well enough for the number that they want. And the second thing, it is technically possible, because it's a whole virus coming from animal cell culture. It is technically possible, and has in the past happened with some of the polio batches back in the 50s, for the inactivation not to work perfectly, and for there to be some adverse reactions, even occasionally some live virus getting through in the past. So there's an inherently higher risk of a whole virus, inactivated virus vaccine. But I'm absolutely certain that they've got all those technical and safety issues cracked. So I wouldn't personally hesitate to receive one of those. They're certainly going to be effective. The second sort of vaccine is the one like the Oxford one and the famous Russian one. Is it called the Sputnik one, I think, which are in use? Oh, sorry, the Russian one's in use, and the Oxford one is applying for release at the moment. And these take just a little bit of the virus genome, the bit encoding the spike protein that I showed you on the surface of the virus, and they put the gene for the spike protein only, and not the rest of the coronavirus, into a safe viral vector called adenovirus, which is another type of common cold virus, if you like. In the Oxford case, it's an adenovirus from chimpanzees. In the Sputnik case, it's 2 different adenoviruses, both human. Probably doesn't make as much difference as people would like to think which type of adenovirus they're putting them in. And those, therefore, are much safer in the fact that you can't generate SARS-CoV-2 from such a vaccine for certain. And the adenovirus they're putting them into has a long track record of safety. They're also relatively, compared with the whole SARS-CoV-2 virus, cheap to make. So I know for the Oxford vaccine, at least, they're aiming for something like £3 cost of goods per recipient, which is quite cheap. I mean, it'd be nice to be cheaper, but it's quite cheap. And that should be a lot cheaper than the like-for-like whole virus thing. The only downside of the adenoviral vectors is that they were specially devised for pathogen to generate immunity. against difficult pathogens where you needed a really good T cell response. So these intracellular pathogens like TB and malaria and HIV and so on. And they aren't really optimized for making good antibody responses, which is actually what you need for coronavirus infections. But as you can see, you do get antibody responses, and they are adequate. Then we've got not yet licensed anywhere, I don't think, and I don't think I've seen any phase 3 trials yet, subunit vaccines. This is where somebody makes the protein, just the spike protein or a bit of the spike protein, and presents it in a certain way to make it a good immunogen. I'm indeed working with one group on such an agent, and we've looked at mouse immunizations with that recently, and it's spectacularly immunogenic. And those with the right sort of systems for production are much cheaper again than the viral systems. So it'll be quite exciting to see what might happen over the next few months in terms of phase 3 trials of those sorts of things.
Sure.
I'd be very, and the safety side of those things is very good. And then finally, I mean, there's a huge range of them, isn't there? Finally, there are you shouldn't ask these questions. There are the RNA vaccines, which of course Pfizer is one. The Pfizer one and the Moderna one are two. Pfizer's licensed already. I'm sure the Moderna won't be far behind because it looks very, very good indeed. These vaccines are a totally new approach, really, in human vaccinology, in terms of widespread use. And I have to say, I got these wrong. So I was predicting 6 months ago when I heard that Moderna were making an mRNA vaccine, that'll be far too expensive, far too unstable, and it won't generate good enough immunity, is what I was predicting, all right? Well, it is expensive, all right? So it's 10 times as expensive as the adenoviral one per patient. But perhaps that's not impossibly expensive. It is unstable, so you do have to keep these things in minus 80 degrees centigrade freezers, which most of us don't have in our kitchens. I don't know, do you have one in your kitchen, Abhi, at home? Probably not. And certainly most people don't have. So
many at Sunway.
Yes, plenty at Sunway. So they tend to had to be rolled out through major centres like Sunway or universities or major hospitals, rather than in general practitioner clinics and so on.
Right.
But surprisingly, they are very effective, and they're generating very good antibody responses and very good protection. And they are certainly going to be safe. So a lot of people are very worried about them because they've not heard of RNA before and so on. But RNA is an unstable molecule that gets degraded really quickly in the body and doesn't hang around. And it's not going to have any adverse safety profile unless something very peculiar happens.
Sure. Thanks, William, for that snapshot. But there's a question by C.K. Liu, who wants to know, out of all the candidate vaccines, if you were asked to choose, which one would you choose?
For myself.
For yourself,
with no expense spared, I would take the current Pfizer vaccine if it was offered to me tomorrow, for sure.
All right.
For sure.
Yes. I think that's also a very good message out there.
Yeah, absolutely. Yeah, because I'm, unlike you, Abhi, I'm at that age range where if I get this virus, it's not so good.
Sure.
So I would say that's the decision I'd make for myself, because I could afford £30 for a dose. But it's not available either for me.
Sure.
Yeah, I can't buy my way into that in this country, which is a good thing. So what I'm likely to have available to me will indeed be the AstraZeneca vaccine. And I would be perfectly happy to take that, knowing that my chance of being protected is not quite as high as with the Pfizer vaccine.
Sure. So there are a couple of questions regarding, can it be used on children? Would the vaccine be prophylactic or therapeutic? From Dr. Kavita Reginald from Sunway University. And I guess, there's really, this debate at the moment, right? Circulating regarding who should be vaccinated first when the vaccines are rolled out. So
these are really difficult questions. Actually, the one question is it prophylactic or therapeutic? That's the easy one. These are prophylactic.
That's right. So my question is, I'm not going to ask you about, which, as long as they, in Malaysia, I'm just hoping that they will start off by, alphabetical order, and maybe then I'll have a chance of getting it early.
B is very far down the alphabet,
No, here we use 4 names.
Oh, so you do.
Abhi, yeah. But anyway, if you had already contracted the virus and have since recovered, would you still require a vaccine? I guess the question is, in terms of bolstering immunity, how does vaccination differ from naturally acquired infections?
So that's a really good question. We don't have enough data on this particular virus to tell us that, but we do know with human coronaviruses generally, although you produce a strong immune response to them, that immune response does wane with time. So you can get reinfected with the same virus in subsequent years, usually a few years downstream, but in subsequent years. So circulating around the world until SARS-CoV-2 came along, there are 3 common plus 1 less common human coronaviruses. And you will frequently, by the time you get to adulthood, have had all 3 of them at least once or twice. So what I would expect, we don't have all the data on this yet, what I would expect is that if you've had recovered from COVID, you probably do have a good enough immune response for a good 6 months or so after that you won't get infected again. But it's entirely possible that you might be advised to have a booster every year or 2 thereafter if the virus is still circulating and still causing the same sort of trouble. And it's generating an antibody response in very much the same way as the virus, but without the danger of giving you COVID.
Yeah. So a question from Vinod from Monash along these lines. Is there any data to show that these vaccines can elicit sterilizing immunity, even for specific individuals? Are there any data out there?
The published data, as opposed to stuff in press releases, is quite thin.
Right.
What I've seen, however, from my site of the Astra stuff, which isn't in the public domain yet, is that there is evidence of in a subset of people elimination of virus in the body, in nasal. So in other words, people become PCR negative fast. But it's too soon to say that. What they call the endpoint of the trial is protection from severe disease, not prevention of infection or transmission, because those are much harder things to measure. Obviously, much to be desired is a vaccine that not only prevents you from getting ill, but also prevents you from making other people infected. And only with vaccines that can do that will we get to so-called herd immunity. And we won't know for certain whether any of the vaccines do that well enough for a little while yet, I'm afraid.
Sure. And there's a question, and I suppose to a certain extent, you've kind of answered it. Quite a number of people on the chat want to know, how long do you think this immunity will last?
Well, the evidence is, because don't forget, it might seem like it's been with us for an awful long time, but actually it's less than a year.
Yep.
So natural immunity, as far as we can measure it in the blood of people who've had COVID, seems to remain high enough to neutralize virus effectively for at least 6 months at the moment. That's what we can tell. What people sometimes forget is, just because the antibody I measure in your blood might have dropped quite a bit, you will have what's called immune memory. So that if you get exposed to virus in the future, and that virus starts to try and replicate in your upper respiratory tract, you will mount a much faster, much more vigorous immune response, which is likely to shut the virus down really quickly. So even if you can't measure the antibody very strongly in your blood now, you will probably make a good enough response to another infection to stop you getting ill.
Great, thanks. The next question, I suppose, is regarding public reception. Quite a number of people are really interested to understand, how do you deal with, the various different sentiments that are out there regarding COVID-19 and the vaccine. And it's obviously very polarizing at this juncture. Even in countries in the UK, I must say, I'm very surprised by the level of hesitancy. Last month, of course, when you hear about Pfizer, Moderna, AstraZeneca, Oxford, and their impressive efficacies, I guess when you think about as a scientist, you think it's amazing that what used to take 10 to 15 years to achieve now has been achieved in 1 year. It's outstanding. But many are also concerned about the safety of these vaccines. And I suppose, when Dr. Anthony Fauci flip-flopped regarding the speed in which the UK's regulatory agency decision to approve the vaccine, now when you see in the social media, it's being used by anti-vaxxers or skeptics as evidence of pharmaceutical industry interference or greed. So in the spirit to roll out these vaccines quickly, whether it was from a public or private interest, have safety considerations been compromised? And we have the Deputy Director at the Ministry of Health Disease Control Division also wants to know, how confident are we about the data predicting these long-term effects and safety?
Well, I think on safety, as far as the RNA vaccines are concerned, scientifically, I would be absolutely amazed if there's a long-term problem with safety with the RNA vaccines. The only issue is going to be effectiveness, and that has been proved to my satisfaction, really. The long-term safety issues will be about the more old-fashioned vaccines, the formalin inactivated vaccines, or potentially some of the cruder adenoviral vaccines. But I would be surprised to see any long-term safety issues with the RNA vaccines. All of the evidence I've looked at and the assurances I've had from people that I trust in the MHRA regulatory authority in the UK convince me that they've gone through all of the stages and used all of the criteria that they normally would. They've done two things. They've expedited the amount of effort they've put into the number of hours people have worked on the committees to review the data. And they've done several of the tasks in parallel that would normally be done in series. But all of the criteria that would normally be used for approving have been approved in the timescale. And the approval has happened under current, I must emphasize this, under current EU law, with an exemption for emergencies under EU law that is allowed to nation states within the EU. So there's no suggestion whatsoever from any reputable sources I've come across that there's been anything other than highest level of probity. And I think Anthony Fauci, who I've known for years, rather regrets his slightly offhand comment, which he didn't really mean, but because it gets taken out of context. And let me tell you, if there had been any bowing to political and financial pressure, it would have been the AstraZeneca vaccine that would have got pushed through fastest, because that's where the most local interest lies. And it hasn't. It's not yet there. It's not yet got the data set required for MHRA to give full approval. And so I think that itself gives you confidence the system is working well. But let me tell you, the long-term consequences of getting COVID-19 for really quite a substantial proportion of those that get infected are non-trivial. And so avoiding COVID-19 infection, I think, it must be people's priority at this point.
Sure. There are a couple of questions from the audience, including Muhammad Firdaus, who asks, what's the public reception of the COVID-19 vaccine in the UK, and has it shifted over the last couple of days since that announcement? And I guess another question, which I guess you can work together, is from Yi Swan Poo, who asked, how do you address anti-vaccines concern? And I think probably can come from your personal experience, but I'm not quite sure whether the individuals you interact in Oxford.
No, I'm very lucky that my job doesn't involve me having to persuade the anti-vax brigade. I'm not sure that I'm necessarily the most tactful person in the world to do it. Really, Abhi. Maybe you in your more public-facing roles are better at doing that. Public sentiment on these things does shift quite rapidly. The sentiment shifted in the polls that I saw over the course of the late summer, early autumn, from a lot of skepticism to a lot of positivity. quite rapidly, and I'm not sure in response to what, to be perfectly honest. But I think what's really important is for public figures that are trusted and that understand what's going on in the science and the regulatory stuff to publicly stand up and take the vaccination if they feel it's safe, and to let people know that, because there's a lot more effectiveness of David Attenborough, for example, taking the vaccine than my advocating it would ever achieve.
Well, I'm sure you have your fans as well.
Smaller fan base than David.
So there's some also very interesting questions. I mean, obviously we have a really heterogeneous group of the audience, and this question is obviously somebody with some biological background. Since the COVID-19, so this is by Yi Si. Since the COVID-19 vaccine is designed based on only the partial region of the spike of the virus, will that be potential to offer broad protection considering HLA differences? So I think this goes, along with the Director General of the Ministry of Health, Malaysia, recently announcing that Malaysia will also be starting a phase 3 clinical trial for a Chinese vaccine.
Yes.
Suggesting that that there will be population differences, and so you would need to have these trials to test them out. So what are your thoughts on this?
No, it's a really, really good question, and it's one that's very, very hard to predict. That's why we do phase 3 trials in so many territories, to try and get a good sampling of the world's population of MHC alleles. And that's why it's important to look at the whole data set from a trial in multiple territories, and not just, for example, not just the small subset of 2,700 recipients in the Oxford arm of the trial, which got the low dose, for example, that was being talked about, because that has a much narrower range of MHCs. So yes, the approval process takes that into account, and it is really important to note that these trials, I think I showed a graphic in my presentation, are going on in territories right around the world, and that's a really important thing to see.
Along that population-specific differences, there's a question from Tay Keng Fu from Sunway University. Professor William James, do you think that the severity of the disease could be due to genetic predisposition of the infected individuals, or is it mainly due to the different strains of viruses?
Well, that I can answer. But there's a third thing, which are other factors. So we know that the genetic variability in COVID-19 is there, and it enables us to track the epidemic quite well. So there's a paper just out today showing they've used that sort of genetic information for their DNA detective work, if you like, to show that something like 33% of cases in Malaysia all stemmed from one outbreak at a religious gathering. Yeah, so that you can tell from that sort of information. But those genetic changes are by and large of no real consequence in this particular virus for the immunity or the pathogenesis of the virus. It's actually a relatively conservative virus compared with, for example, influenza or HIV or HCV, which vary a lot. So we have zero evidence as yet that there are more pathogenic and less pathogenic variants of this virus. However, we do have quite a lot of evidence that there are a host genetic susceptibility variations that are just things you acquire from your parents, and they can, for example, be in those HLA regions we talked about a moment ago. They can be in your interferon genes, which is a defense gene, and one or two other proteins that are involved in the virus replication. So we're beginning to understand those a little better. But the biggest variables that determine how sick you're going to get appear to be your age. So being older, being male, those by far the biggest risk factors. And then the next ones are things like diabetes, which I know is a very big problem in Malaysia, isn't it? So comorbidities like diabetes and being overweight and high blood pressure and things like that are very significant associates of serious disease. Another good reason for laying off all that sugar in your coffee, guys. And socioeconomic situation.
Yes.
As always with so many diseases, it's a really big driver of pathogens. So if you live in poor circumstances, you have poor nutrition, you have poor mobility, you're at higher risk. Sadly, multi-generational families are also at higher risk because of the transmission chances. But yeah, so those factors are there.
That's right. And of course, ethnicity. So that's one thing going for you, William James, when it comes to protection.
Agree about this. I don't think it's anything to do with ethnicity, because I have ethnicity as entirely a social construct and nothing to do with genetics.
Absolutely. And that's where I was going to go to Faisal Arif's question. Okay. With cost and distribution consideration, how long will it take for the world to go back to pre-pandemic normal?
Well, I think it'll be a new normal, won't it, Abhi? It won't be entirely the same. And I tell you, the liberation of being able to do so many of my things online, as opposed to having to go to meetings, not having to trundle down to London to meet people, but be able to do it on Teams or what have you, is fantastic. I even do my tutorials not in this room. My students come to this room, but I teach from home. And it works better, would you believe? I mean, it'd be nice to meet up with them occasionally, but it works better than face-to-face. So we will change. Everything will change. But the serious question is, when will the vaccine or the vaccines have gained sufficient traction in the world that transmission has now died away and we can return to a more or less normal life? Well, it's not going to be before the end of this year. I don't think it'll be before spring. I think it'll be something like, spring is not a sensible term in Malaysia, is it? I don't think it'll be before March, April. I think it's more likely to be July, August next year.
Right.
Yeah, but I think it depends on what gets approved. If the cheaper, easier to produce vaccines like the AstraZeneca ones do get approved, they can be rolled out at the sort of level that can do that for us. And the efforts that are seriously put into in international agencies to make sure that these vaccines get distributed as equitably as is practicable around the world and around communities in different countries will make a huge difference. And I think that vaccine will certainly be available soon after Christmas in the doses needed, if it's approved. If it isn't approved, of course, then we'll have to rely on the RNA vaccines, which are not so easy to scale up, but still possible.
Sure. It looks like there's still some biologically related questions. So I'm going to segue back again before we go to the big picture.
Yeah.
The question is, if immunity lasts for 6 months, will the virus mutate after vaccination?
Well, of course, the virus will mutate. But the good news is that the 3 or 4 endemic coronaviruses that go around human populations have not significantly mutated as far as their antigens are concerned, as far as your immunity is concerned. They're not like flu in that respect. They remain very conservative. So the likelihood could be different this time, but the likelihood is the vaccines that are made and being approved now will work long-term for this virus.
Great. And Sangeeta has a question regarding the side effects of the Pfizer vaccine. So I know it might be unfair to you, but I guess the question is, do these vaccines have a higher chances of exacerbating mainly T-cell mediated autoinflammatory conditions? And is there any data to suggest that?
No. Of course, you can never exclude very rare events. When you're deploying billions of doses around the world, there will be rare events for certain. Somebody with a very unique combination of extreme sensitivity to this and a predisposing factor for that. But at the level of numbers of serious adverse events per 100,000, I don't see any reason to, in the data that I've seen so far, I see no reason to suspect that these RNA vaccines, which are just getting approval, have anything in them to worry about whatsoever in that respect. Of course, if you're an individual that has severe underlying health issues, you should always take advice from your medical practitioner about this.
And I suppose, sorry, William.
No, not from Facebook.
Right.
Just get it from somebody who knows what
That's right. But I suppose that's where the problem is, because often anecdotal evidence and this individual instances, presents itself as data, or probably a bigger problem, and that's part of the problem in terms of communication.
This is, I think, the hardest thing that I have in science communication is that we are so tuned now in our highly networked world to be found for us, brought to our attention, the extraordinarily rare but very, very tear-jerky, that's extreme example. Yeah. We tend to think of those as somehow being representative of a large thing, and many of us are not trained. Now, of course, you and I are trained to do this, but many of us are not trained to think in quantitative terms over several orders of magnitude. Yeah. So what we're thinking about here is balancing up risk between the risk of an adverse effect that might be 1 in a billion effect of a vaccine against the several percent mortality rate of this virus and the many percent adverse long-term consequence effects of this virus. So that's a huge difference in risk and really not comparable.
Sure. And coming back to the skepticism, there's also, I guess, to a certain extent, this whole interest in how remarkable the science was in developing this vaccine. There's also concern why, many diseases that perhaps are in the poorer countries or LMICs are often neglected, and they call them the orphan diseases. And I think Anjan and Rani's question is, why are there no vaccines for malaria and AIDS yet?
Actually, those are two really good examples. That's a fantastic question. For those two diseases, it isn't for want of effort. In fact, throughout my career, there's been vast amounts of effort to make an HIV vaccine with nothing really to show for it. And not because of the lack of money. I mean, the very large amount of money went into it. It's because it's not you can, if you want to look at my article in 1987 in the journal Vaccine, I explain all the reasons why HIV is going to be a very tough nut to crack, and we don't have any good leads at that point. We still don't have any good leads. It's just intrinsically a very, very difficult virus to do anything about because of its biology. Whereas coronavirus is the other end, the easy end we know we can make vaccines against the coronavirus. Malaria is another example. Now, the Oxford team, in which I have no vested interest, have already made a similar style vaccine, which has been in trials against forms of malaria, and have been of mixed outcome, because malaria itself is a very, very difficult pathogen to make a vaccine against. And as you know, Bill and Melinda Gates put a great deal of money into trying to develop vaccines for malaria. But you're right, there's a tendency to focus the R&D dollars on diseases that affect the rich world. But this virus is no respecter of wealth. It hits the whole world. So I think we're all in this one together.
Sure. Before I ask you the last question, and we've only got 2 minutes left.
Oh, no.
I know, time has just flown. We'll just have to invite you again, and I'm sure we will. Muhammad Azri's question is, what are your estimates in terms of when do you think the next pandemic is going to occur?
After I'm dead.
Well, I hope you'll be alive if we have the next pandemic,
or
when,
that's a difficult
so that you'll be able to find the solution.
First, I think.
we need the brains, and I guess we probably the reason why we're all here tonight, the reason why I'm sure you lose a lot of sleep, and as you rightly pointed out, the billion of individuals, whose lives have been affected, by the economic, and various different, sort of losses, is just staggering. And I kind of joke that the Oxford English Dictionary at least will benefit from new words like COVIDiots and Zoom bombing and and quarantinis and what have you. But jokes aside, we are also glad that we have brilliant minds like yourself working towards finding solutions, and we know how COVID has affected. In fact, there's a verb, COVIDized our lives. So how has this whole experience changed your perspective, and perhaps even Oxford's perspective, on how we need to improve humanity's preparedness for the challenges ahead?
It's a really good and deep question. It's a pity there's only 0 minutes left to answer that one. But
it's okay. I'm sure
let me just address it as briefly as I can. Obviously, it's disrupted all our lives. I mean, mine as well as people in my lab and my students and the rest of it, in very serious ways. But there are little silver linings, and the little silver linings are that people are actually understanding that public health is a matter of public good. It's not just a matter of private interest. That, for me, is a really important lesson that people need to understand. It's not just about my needs. It's about actually you can't detach yourself from society in this. And I think in the West, at least, that's been a lesson that's been painfully relearned recently. The second good side of it has been the extent to which my colleagues, not just around Oxford, which, as you know, is a place that specializes in bitter infighting and backstabbing and all the rest of it, but actually around the world have really put aside all their personal rivalries and teamed up to share best practice and best techniques and so on to make a really joint effort to tackle this virus. And actually, the Pfizer one is a really good example of that, where you've got a couple of Turkish origin in Germany that developed this vaccine approach that's been picked up by an American company as first license in the UK. It's a massive international effort across all teams, and if we can keep a little bit of that collaboration going for a few months after this all gets better, that would be quite nice.
Yeah, I think you're absolutely right, because there are many things there that completely changed our lives. But having said that, the one hope we have is this connectivity, and I'm sure the audience today can agree that William, having you with us for the last 1 hour just makes us feel connected, makes us have hope, and also ultimately realizing that every one of us have an individual responsibility to collectively come together and really overcome this crisis. So on behalf of the Oxford Society, I would like to thank you, William, and also to the audience for staying up this evening. And in fact, you were competing with the Nobel Prize lecture that was happening at the same time. So,
what a shame.
Don't worry, they're all recorded, and including this lecture. So this session, and I look forward to engaging you further, and hopefully we'll have you at Sunway sometime very soon, William.
I'm sure we will.
Thank you. Back to you, Emma.
Bye-bye.
Thank you. That was amazing. I'm definitely going to have to listen to the replay on our YouTube channel, because I was so busy with all of the questions that I know there were lots of answers that I wanted.
They were good questions.
There were great questions, really great questions. And, I especially appreciated the roundup that you did of all the different vaccines that are out there. And also,
I didn't go on too long with that one, because that was a difficult one to do short, isn't it?
But I think that's a really important one. I mean, I'm not a bioscience person, so for me, I thought that was very important. And also, your view on what we do know and what we don't know, because that's the other thing. People have all these various theories and they say this is fact. So I really, as a layperson, appreciated you saying what we do know and what we don't know. And also, basically, the work that you guys are doing. I imagine for the last 10, 11 months, you've all been working flat out for the rest of us. So I think a lot of people do recognize that and appreciate that. immensely. So yeah, thank you. It's a great job. So thank you so much for coming on. That was amazing. And thank you to Melissa for helping.
Thank you, Melissa.
Thanks, Melissa.
Thank you to Professor Abhi for moderating us.
Brilliant. You're natural.
Thank you.
Starship.
Yeah, that's a lot of questions. You were fielding them all together. So thank you.
Yeah.
Thank you. That was a great job.
I've done that. It's really hard.
Well, Melissa and Emma, you made my life easy. You had also summarized them, and they were quite systematic for most part of it. So I found it quite easy, and I knew that whatever I fielded to you, it's always going to sound really good. So, William, thank you.
So just quick before we go, don't forget, guys, on Wednesday, 8 p.m, we are having a fireside chat with YB Said Sadiq MP for Moa and Sharil Sufian Hamdan, who is the Deputy Chief of UMNO Youth. So see you guys on Wednesday, 8 p.m. So one final thank you.
You're not expecting me, are you, Emma?
No, you're clear. No, you're more than at your supper, and we would love to entertain you when you next come over
when I visit properly.
Would be great. So thank you so much, Professor William James. That was amazing. Thank you.
Thank you.
Hey, good night, everybody.
Bye-bye.
Bye. Good night.
Bye.
Bye.